Hello students, we are continuing our topic cell cycle. In yesterday's class we have seen that what are the changes which is happening to the cell in interphase. Okay, interphase means the phase just before the division. Interphase is divided into three phases, G1, S and G2. Okay, in G1 what is really happening? The cell after the division, a newly formed cell is just entering into the G1 phase and the growth of the cell is taking place here in the G1 phase. Okay, the enlargement of the cell and the formation of the cytoplasm and the protein production, RNA production, and ATP is also producing here in G1. So all these things are happening in G1 phase, and after G1 the cell is having two options. One option is that it is just entering into the S phase. Okay, otherwise the cell can withdraw the cell cycle to a resting phase, and otherwise we can call it as a G0 phase. Okay, so a newly formed cell is entering into the G1 phase and it is attaining the growth, and normally the cell is going to the S phase, and some cells are not going to the S phase, but it is going out from the cell cycle to a resting phase, and we can call that phase as the G0 phase. See, the resting phase means the activities are taking place. What are the activities which are taking place in a normal cell? It will take place, but that cell is not going for the division. That is the only difference. Okay, so after G1 the cell is entering into the S phase. In S phase, what are the changes? The replication of the DNA is a major one, and the centrioles are also replicating in S phase. And afterwards it is entering into the G2 phase, and in G2 phase also the production of protein, production of RNA and ATP will take place. Afterwards it is entering into the division phase, otherwise the mitotic phase. Okay. So today we are going to discuss about the various steps of the division phase. Okay, otherwise the mitosis. So mitosis or the division. So see, you have to recall that what thing is going to divide. The cell is going to divide. And what are the two major parts of the cell? Nucleus and cytoplasm. So firstly, the nucleus of the cell will divide, and afterwards the cytoplasm will divide. So that in the division phase there are two major steps. The first one is karyokinesis and the second one is cytokinesis. What do you mean by karyokinesis? Karyokinesis means the division of nucleus. Okay, karyon means nucleus. Division of nucleus is taking place firstly. Okay, so the division of the nucleus is not a single step process. Okay, it is really taking 4 steps. Okay, and what are the divisions in karyokinesis? Otherwise, what are the steps in the nuclear division? The first one is prophase, second is metaphase, third is anaphase, and fourth is telophase. Okay, and after this telophase, otherwise prophase, metaphase, anaphase, telophase, all these things are finished, means the division of the nucleus is done. Okay, the nucleus is divided into 2, and after that the cytoplasm is dividing, that is cytokinesis. Okay, and we are going to look the prophase today in our class. Prophase means the first step of karyokinesis. Okay, so this is a cell that has just finished the interphase, means this cell is ready to divide now. Okay, so in this figure you can see the prominent nuclear membrane over here, and there are so many pores over here. That is a nuclear pore. Inside the genetic material will be there. Genetic material means the chromatin thread. It is in the form of a thread. Okay, and what is really happening to this cell when it is entering into the first step of karyokinesis, otherwise prophase. Prophase, 2 stages are there for prophase: early prophase and late prophase. Like that we can divide the prophase itself. Okay, so what are the changes to the cell when it is entering into the early prophase? See, in this figure you can see that the nuclear membrane is very much prominent. You can clearly observe the nuclear membrane over here in the after the interphase. But when it is entering into the early prophase, the nuclear membrane will start to dissociate. Not only the nuclear membrane, See the endoplasmic reticulum, that is also having membrane, and the Golgi apparatus membrane. All these membranes will start to dissociate in early prophase. And here the genetic material was in a thread-like appearance. It was not condensed, and the condensation of the chromatin fiber will occur in the early prophase. So in early prophase, the chromatin fiber will condense, and we can observe the chromatin fiber as in the form of chromosome. Okay. So the appearance of the chromosome will get in the early prophase. And afterwards, see the centrioles are, actually the centriole has been divided in the S phase itself. Okay, it was the division of the centriole was a feature in S phase. It was already divided, and the divided centriole will start to move from this pole to next pole. Means into both these poles, one centriole will be there. Okay, one of the centriole will start to move from this pole to the opposite pole. So these are the changes that you can observe in the early prophase. What are they? The nuclear membrane, endoplasmic membrane, and the Golgi apparatus membrane. All these membranes will dissociate. That's the first one. And second one is that a centriole will start to migrate from one pole to the next pole, and the chromatin fibers will condense and it will form the chromosome. These are the major features that you can observe in early prophase. After finishing the early prophase, the cell is going to enter into the late prophase. See, the centrioles will start to form small fibers from here itself. Okay, it will be prominent over here in the late prophase. In late prophase, you can see that. See, the centriole is actually making two types of fibers: small fibers and long fibers. It is making. Okay, the short fibers. Okay, these short fibers are known as the aster rays. Okay, what is the name of this one? The short fibers are known as what? Aster rays. And aster rays and the centriole, these together is known as aster. Okay, so what is aster? Aster means the small fibers which are produced from the centrioles, and that is known as aster rays. Aster rays and the centrioles, these together is known as the aster. And the long fibers which is produced from the centriole is known as a spindle fiber. Okay, what it is? Spindle fiber. And the spindle fiber will start to attach to the centromere of the chromosome. Okay, it will start to attach to the centromere of the chromosome. Centromere means what? Centromere in a chromosome, there is a common point for the sister chromatids. The sister chromatids are attached to a common point. See, here you can see that a common point is there for the sister chromatid, and this point is known as what? The centromere. So the spindle fiber will get attached to the centromere in the late prophase. Okay. And the dissociation of the nuclear membrane was started over here, and it will get completely disappear here in this late prophase. In late prophase, you cannot see the nuclear membrane, and the nucleolus also will disappear in late prophase. Okay. So these are the major features that you can observe here in the late prophase. Means you can see the long fibers, otherwise spindle fibers over here in the late prophase, and the spindle fibers will start to attach with the centromere of the chromosome, and the nuclear membrane and the nucleolus will get dissociate, otherwise not dissociate, disappear. Okay, you cannot see the nuclear membrane and nucleolus here in the late prophase. And you can see the chromosomes are in different levels over here. Okay, one chromosome is here, one is here, one is here, one is here, and all these things are not in the same level. So the next level or the next phase of the division, what is really happening means all these chromosomes are going to be come into a particular level. Okay, so how it can be possible? This spindle fiber, see, this much length is there for this particular spindle fiber which is attached to this chromosome. The length of this one is only this much. So what is really happening means the length is going to be equal. Means this spindle fiber is going to contract. Okay, It will just condense, otherwise it will contract. So that what happened? This chromosome will start to move over here. Okay, like that the contraction of or the shortening of the spindle fibers will start to it will start here in the late prophase, and after that in metaphase all these things are, all the chromosomes are coming in a central level or in the similar or equal level. That we are going to see in the next class. What is the changes which is going to happen to a cell in the metaphase, that we will discuss in the next class. Okay, so that's all for today's class. Thank you.